be talking about de-identified clinical data repository, which I will call DCDR because it's a mouthful. And then also, along with that, infectious disease repository, and you will know all about that. So the way I will do it is try to introduce you to the project by kind of year chronology, what's happened in the beginning, how it has changed over the years, and then what is the steps forward. So I can give you an overview and kind of details of what's going on. In the beginning, I was not part of this project because it started in 2011. However, these two projects had slightly different ideas where it originated. DCDR had an idea to provide researchers with easier access to some part of clinical data. And IDR wanted to create some sort of infectious disease repository to advance their research and QI. So these are slightly different ideas, but you could see they're kind of at the core to provide researchers with some data and to advance research. So a little chronology on DCDR. In 2011, March, I believe the person who started this is Nick Anderson, who is not with us right now. However, the process started in March, and there was the approval process, and there was IRB by Nick, which is slightly different right now, but it was approved. In November, December, minimal data set was loaded by Tony, basically demographics, diagnosis, and disposition, I believe. And then they had a focus group where feedback was really positive. Everyone says, give me more data, or we really love this. So the project was supposed to continue on from there. For IDR, Bob came to Tony with this idea of creating a repository. So in April, Bob and Tony wrote a little initial pilot grant application to try to get funding for this project. He was rejected in May. However, they applied for second pilot application in December. So the project is continuing on. In 2012 is when the development of these two projects started. So until September, the CDR became a little bit quiet, and I will explain why. I joined the project in September of 2012. In October, the more data started being loaded, and a project came out of that, which called DHOME. I won't explain unless you are interested. In November, December, I continued to load the data so we can hopefully launch in following year. IDR, great news. In June, Bob Harrington pilot was awarded so he can start his project. In October, now since the project is starting, we need IRB. Of course, research data requires all the IRB. So Bob and Tony wrote an IRB for the repository of infectious disease. In November and December is when the IRB for the IDR was approved. So now the project is happening real in IDR as well as DCDR. I want to take a closer look at what the aims of IDR was so that you have an idea of what needs to happen. So grant aims and IRB aims is to generate a library of data queries and from the EMR and establish the UW infectious disease database, which now we're calling a repository because that's where the data and queries are stored. And the second was to have comprehensive clinical data on patients that are suffering from a variety of serious infectious diseases, focus on infectious disease, and to use these data to perform QI and observational research. So these were IDR aims. So what were kind of envisioned deliverables? One is to enable the faculty and fellows to define cohorts. So they need to figure out what kind of cohort is possible before they do any kind of research. And once they have these cohort generated, they wanted to get a longitudinal data set. So whatever data set that's needed for research. So these are the actual sort of bullet point deliverables. 2012, what were the challenges? I mentioned that DCDR did not move forward until September. There was absolutely no one who had time to work on that. So how it was solved is just additional personnel. That was pretty simple. IDR. So these were two things that were deliverables, right? So what was the challenges in 2012? There was this thing called problem list, and I will explain what that is. And there was a problem with the problem list. In next slide, I will explain what that is. And then how do we enable users to define ID cohort? I mean, we have absolutely no researcher access to clinical data at this point. So how do we make that happen? At the end of 2012, there was kind of no idea. We're still investigating. So let me explain what the problem is with problem list. So Bob's vision of defining cohort is that he wanted to use physician-provided diagnosis for infectious disease because the ICD-9 codes entered by billing he thought was notoriously faulty, and he didn't want to trust that. So he wanted his fellows and his attending doctors to use what's called problem list on ORCA, Cerner, EMR, to actually go in every time they see a patient, go in and enter physician diagnosis. This person had this disease and in this area. And there were 2 items that need to be entered on problem list. For example, someone had to choose infectious disease organism, fungi, and then Aspergillus flavus to determine that this person actually had something. What was the problem with the problem list? We had 3.3% of entire infectious disease note people who had physician-entered diagnosis on problem list. And it was even lower on UW-MC. It was 1.3%. So there was 3.3% and 1.3% of physician-entered diagnosis, even after Bob trying to get fellows and other doctors to use this thing on EMR to determine diagnosis. At this point, end of 2012, Bob and Tony realized that it is impossible to use problem list to determine the patient had some sort of infectious disease because we only have 3.3%. Now we have a problem with the problem list. So what do we do now? So now we are in 2013, last year. So DCDR is steadily moving along. We had version 2 launch planned in February with additional data variables that was loaded the year prior. IDR, now I have joined this project in February. And then with discussion with Tony, Bob, and I, we came up with solution to the 2 problems we had prior. So what are those solutions? How do we determine ID cohort without the use of this physician diagnosis that was a problem? We go to the data. As in, Bob realized that if we look at microbiology lab results, if we get the organism and where it comes from, we can pretty much predict what the disease that is. So in order for you to determine your cohort for, I think, bacteremia, I guess you would say Staph aureus in blood, for example. That would pretty much narrow it down. So it was good enough for cohort identification, which meant we needed 2 main data from microbiology results. One is the culture description, all the list of organisms. The other one is specimen description, as in it's important where they're coming from, whether it's urine, blood. It doesn't matter, it's on your surface because a lot of people might have bacteria on the surface. That's not really infection. So, and in addition, of course, in order to build cohort, you need visit detail, which hospital they came in, what date, all the demographic, and whether infectious disease notes were written because they want to know if someone wrote a note on it or not. And some of the susceptibility to medicine for these microorganisms, and etc. So that was our solution. However, there's also challenges every time you find a solution, which is microdata is not very structured. It is structured, but not completely. If you can see, these are 6 lines of things that we get in one test result for microbiology. So you have, and this is just 2 columns out of that we would focus on. So we have specimen description, special request, culture description in 3 different lines, the status, and the list goes on. But I just focused on this because these 2 boxed items are what we're actually looking for. So we need specimen description on where it was obtained, as in blood here is a keyword. We don't care if it came from rice subclavian. This is from Bob's comment. For culture, we don't care about broth. We care about what organism Staph aureus. So we're trying to parse out what's necessary in the culture and specimen description. And some of the problems, we're doing text searches on these strings. We have results that are negative, not present, all kinds of ways you can indicate that's not a valid result. For example, there's duplicate, reorders, wrong tests, and sometimes they put multiple organisms in one test result. So how do we get around to that? This part is a little bit informatics-y and maybe not as exciting for some of you. But so first we filter all the invalid results, as in these are literally text of all the invalid results that we collected using some of the things that other people developed. Filter all the negative results. Thankfully, many begins with no and negative. There are some not found type of results, but that was minimal. And then we identify search strings so that we can say, hey, this entire string, you can tag it as Staph aureus. This entire thing we can tag as Staph aureus. So we're basically searching for organism names in specimen. And now we also have to identify these items in the specimen description, like blood, arm, fluid, knee. We have done the text searching, and I will explain a little more detail how the result ended. But the next question was, how do we get the actual researcher to go find their cohort? So that's when we realized that, hey, we could merge DCDR and IDR together. DCDR provides a query interface where researchers can go and search by themselves, and IDR needs that. And DCDR basically enables researchers to go in and click on the category, drag and drop, and click and say, hey, I have 512 people who can be my cohort. And this is without individual IRBs, because we have written the IRB for DCDR to cover anybody who has signed the document to be able to access it, because it is de-identified. The issue is DCDR is not the easiest tool to load data into, because it requires certain things. For example, it requires a categorization that researchers can search from. And we need to have two different categorizations. One was the specimen, the other one was culture, because they need to search on both. For culture description, it wasn't bad because all the organisms were in genus, species, and the lab system actually had a list of culture descriptions that a lot of people do a pick list from. So for culture description, we organize it by using the organism, genus, and species, and then, of course, text parsing this culture description to put the data into these categories. On the other hand, specimen description was a complete mess because there was no lab categorization available to us. So these nicely laid out specimen description that you see is result of Bob and my hours of parsing and categorizing. So we took 3 years of specimen description text, completely tokenized it into each word. And then I determined how frequent these words are. Some of them were kind of useless words like and of and all that. So we removed that. And then Bob went through these 1,000 lists and organized them into categories that make sense for clinician. So this took probably like a month of categorization. In the mid-2013, we merged 2 projects. So we decided to insert all the microbiology selections into DCDR. And then we presented our DCDR/IDR tool to the infectious disease section, and they were quite excited. And they're like, oh, this is great. We can train fellows and researchers and go look at this tool to find their cohort and do more research and QI activities. In July, I presented my incredibly detailed microbiology text processing at the I2B2 because DCDR is using I2B2 interface. And ITHS and Infectious Disease Division decided to commit to move this project forward together. And Infectious Disease provided, I believe, about $20,000 per year for researchers to come and actually use this tool, determine cohort, and get the data from us. In August, we had to reconfigure the IRB a bit. The initial IRB was approved for somewhat smaller set of data, so we expanded with some discussion with IRB to expand to all data that is possibly be able to load it from Amalga, which is our clinical data repository, as long as we de-identify them. So it was a nice IRB success. In September and October, we started the DCDR IDR training sessions. And you'd ask, why do we need training sessions? After interacting with clinicians and after the focus group 2 years ago, we realized that, yes, DCDR is very intuitive for a lot of informaticians to use. It's drag and drop and or comments. However, clinicians are still having some trouble with these reasoning process. So having the training classes really helped them to get a head start instead of getting frustrated after just initial entry. And in November, we made some enhancements to add HIV data elements. And in December, Bob requested a QI project through DCDR, which I will explain in the next slide what that was. And also in December, I did a mini collaboration with Maliha where I tried to get the assertion on yes or negative, yes, no, negative results on the specimen description using her NLP assertion tool. And it seems like it might work out for the microbiology text, and we submitted abstract for that in December. What was the QI project that Bob and the ID section was somewhat excited about? Was basically he was looking at HMC patients with Staph aureus and endocarditis and seen in 2013. He wanted to find out first how many people of this cohort had echo done. It was recommended. And then he realized out of about 19 patients, only 2 were recommended by the infectious disease doctor to get this done, even though it is a recommendation by American Medical Association. So this generated a whole lot of discussions during the section meeting as to what's going on, what's happening. Maybe that's not necessary. Maybe we need to collaborate with other departments. So that was quite exciting. And then second one is of those cohort, how many patients receive gentamicin. So what were the 2013 challenges? We talked about this problem with the problem list and the user defining cohort. So we solved it by micro lab data, query interface, and building ontology, which took quite a bit of time, but once it's done, it was great. So 2013 IDR challenge, DCDR/IDR challenges. How do we enable users to effectively use this tool? So that's why the training and support was very important. And initially, the training was about an hour and a half, including all the introductions and detailed steps on how to use it. And we felt like that was a bit difficult for doctors to come for an hour and a half, sit through the entire session. So we streamlined the training process, put half of it on video, and then we just have them come in and try to struggle and solve problems that we give them like a quiz. And we had that session in November, and everyone thought that was really helpful because those of them who watched the video actually was able to play and try to struggle and ask us questions while doing that. And of course, there's always the data enhancement. Please give cohort identification data that we need, you know. I guess the pulmonologists want the respiratory data and orthopedic doctor wants some sort of surgery data. So there's always that struggle. And next step is to do evaluation, as in I'm trying to speak to people to figure out, like, was it really easy to use? Was training helpful? What else will make these more useful, as in you actually go in and do things on it? And also the last 2 bullets are kind of I guess, chicken tongue black bullets. But there's a lot of coercion involved from faculty like Bob and Tony and I to try to get users to use the system. Remind them that we exist, remind them we have done enhancements, remind them there's next training, please have the other people come and sign up. Because this is kind of like on their second thought brain, because not all the doctors need to do research. So in order for them to actually come and use, unless your fellow's dying to do research, it's kind of, you gotta keep poking at them to try to come and see it and advertise to other people. So I'm just going to give you a current status. From the 3 lists, we have added these huge list of elements. If you want a copy, it's on the website. I'm just giving you. And we are continuing to add small little modifications. The current status is that we have right now processed to have researchers identify cohort on the interface. And we have a website for information and feedback, very generic and very low maintenance. But, and then a way to have people request data and return it semi-automatically, because it still involves us. In terms of innovation piece, basically trying to get those categorization of infectious disease and text mining the data, which we have done that for starting 2010, so that's quite significant processing of text. There has been a lot of training done. So far we had only 3 sessions and 30 users, but it's from all three different institutions. And these are right now mostly senior faculty. However, we have now streamlined the training, as in you listen part of it at home and you come in for half an hour. We're hoping to run sessions every month or so. So in terms of presentations and posters, the conference poster I have mentioned. We have done a couple presentations in infectious disease, and I think that's going to continue. I have done a poster submission. It was more like an abstract late-breaking session for AMIA CRI, which was the NRP replacing possibly what we're doing with the negative specimen parsing. In terms of collaboration, right now we have infectious disease division and proof of concept natural language processing. So what are some of the breakthroughs which I thought was really cool in 2013? When we first presented this tool and did a little demo at the division to get their support, there were a lot of people like, wow, this is cool, including, of course, Bob. It was always excited, and King Holmes, and a lot of attending level doctors. And then first 2 training sessions, everyone had these light bulb go up at the end of the training. I believe we recorded some of that. They was like, oh, this is great. We got to give it to fellows and all that. And the QI project presentation was, I think, quite a success. Out of 60 minutes, we had 10 minutes of discussion. So considering that some people had only 2 minutes to speak in that section meeting, it was quite significant. And we keep getting the referrals to DCDR training just by word of mouth. That's how we're feeling. We're not advertising even right now, but we have at least 5-6 people per month just getting word of mouth. So what's planned for this year? Initially, January training session, actually we're planned for February. And then I will be talking on 24th at Infectious Disease Fellows Day for half an hour to give them the training for the tools. And then we'll be developing some IRB templates for infectious disease research, so it will be easier for people to fill out the IRB requirements. Bob is going to release the request for project with the division funding that we talked about to the fellows and other people right after the fellows training date, so we'll get the actual projects for research. I have an evaluation interview set up with one of the users, one of the fellows who are actually actively using, and I believe he's in the process of submitting IRB for research study. And we have another one who just emailed us who will be trying to request data this month. So rest of the year, we'll continue to do training. We'll continue to work with infectious disease divisions. And then I'm hoping to, again, lots of coercion to do some evaluation research on research. So moving forward, DCDR is supposedly sort of a part of the ITHS initiative, the ITHS badly named. So we will hopefully do an evaluation research on research and publish the result, hopefully, with a lot of coercion on faculty from me. And we hope to collaborate with other departments, possibly incorporate NRP, and maybe incorporate data from other sources. These are all big, grand visions. And I just want to briefly mention what we couldn't do without. If you don't have help from the actual user of the system or someone who has a vision, like Bob, with huge enthusiasm, this project is really difficult to do. Because we spent many, many hours of discussion with Bob to move this project forward with his vision, with our attachment of it with the CDR. And of course, department support and support from ITHS. And this is my last slides. So this is sort of a vision slide, as in continuously innovating how to enable research data access. It started with just manual chart abstraction, basically needle in the haystack, or you ask the lab department to give me a list of 3,000 patients who had this test done, and then you go through all the test results, and then you go to their chart, and then you look at their diagnosis, and then you go look at their echo from their diagnosis. So this is like hours and hours of work. From there, we have moved on with the clinical repository to our consulting team doing queries. So now they can tell us what they want, but it also requires consulting negotiations, and we spend a few hours looking for data, a few hours writing queries, and it costs them money to do so, so they can't get exactly what they want. DCDR and IDR is sort of halfway service, as in cohort department, you can go and do it on your own. It doesn't require our time. We don't charge them for identifying cohort, which in this section we could, custom queries we could have. However, to get data, they still need our help because access to identified data is through IRB Unlimited. And the ITHS.com, badly named, is the goal here.